My name is Daniel. I'm not going to show you like proper results. I'm going to show you more what we're doing so far and where we're going. Because basically we're part of work package 6, so we were at the end of the line from all the other work packages doing comparative analysis of the different data. In my case, I was more focused on comparative analysis of non-coding RNAs, okay, and a bit more in comparative analysis of genomic relation between sexes and tissues. So there is a lot of partners involved here, directly or indirectly, but basically the non-coding RNA task is the one I'm going to show more and has been coordinated by our colleagues in Crete. So probably you all know, but just a bit of introduction, non-coding RNAs are supposed to be RNAs that do not codify for proteins. Well, at least the majority of them. And they are broadly, classified in small ones, that is under 200 nucleotides, including like the classical ones, ribosomic and tRNAs, also microRNAs, as our colleague just explained. And the long non-coding RNAs is over 200 nucleotides. There's different classifications, okay, depends on the structure, the action, the location. But mainly, what we are working in here is the long non-coding RNAs that are called intergenic ones, okay? That's the one I'm gonna speak about, the intergenic ones, between the genes. There are many different ones, but that's the ones I'm gonna talk. These long non-coding RNAs, they have many different functions, okay, depending where they are, but basically what they do is regulate the transcription of the genes. Could do that directly in the nucleus by interacting with the DNA, like helping to open and close the chromatin. Sometimes they work as a scaffold for proteins. Sometimes they are a decoy for this same protein, so they can prevent or promote to open the chromatin. Sometimes they guide them, so they interact with them, and sometimes they respond to stimuli. In the cytoplasm, they also have a very important function. Sometimes they act as sponges for microRNAs. It's very useful because they are used as decoys. And sometimes they can interact directly with the messenger RNA, either stabilizing the mRNA or helping to degrade it. So they have very different places where they can work. Yeah. So in general, that's a bit old, but it's still holding. In general, when you go to mammals, the number of long non-coding RNAs is quite big. It's really, really big. I mean, if you take a look into the Ensemble latest genome annotation of the human, you can see that 29% of the genes are long non-coding RNAs. So it's pretty high. Okay? It seems that fish is a bit lower and much lower in invertebrates, but in zebrafish we have at least roughly 1,500 long non-coding RNAs that represents around 5% of the genes that are annotated. In the AQUA-FAANG consortium, if you take a look at the species, carp and salmon, they have a much higher number of long non-coding RNAs. It goes up to 15, 18%. These, the sea bass, the gillhead sea bream, and the turbot, they have similar percentages than the zebrafish. And the rainbow trout is the only one that seems to have a lower number of long non-coding RNAs, at least in proportion, not in number, but at least in proportion compared to the carp and the salmon. It's a bit strange, but well, that's what it is. So we suppose that this higher number is due to the whole genome duplication, okay? But that needs to be seen a bit more. So in here, I was trying to do a comparative analysis between the 6 different species, okay? What is a problem? Because initially, many people, what they use to do these comparisons is the sequence. So here is an example of that using different types of flies and mosquitoes. And in here, just don't bother too much about the numbers, but here are the numbers of similar non-coding arrays between the different species. And you see numbers are not too high. It's about, in this case, about 160, 150. But look at that. They are very close related species. They are the same genus. In our case, we're talking about species that they're not even in the same group. So they're really, really far away. So the question in here was, if we want to look to conserved long non-coding arrays, should we look into the sequence? Or maybe should we look to regulatory regions? Because some authors, what they say is what is conserved is not the sequence, but the region that regulates their expression. Or some people, they say that what is actually conserved is the structure, not the sequence, because if you take a look at the long non-coding RNAs in general, the sequence doesn't seem to be very, very conserved. Or maybe what it's conserved are the interactions. So what we're doing is trying to tackle all 4 of them. It's a bit of a work. Here is just some examples of a crude analysis of the long non-coding RNA expression data that has been generated by the consortium so far. In here, I'm not distinguishing between sex or anything, okay? Just taking a look, or mature and immature. Just taking a look what is in there. And here it shows the long non-coding RNAs that are expressed in average, over 10 TPM. So normalized counts. We can see that embryos, there's a bit of a range of different long non-coders expressed. CARP has a lot of them. Here's as percentage. And here the long non-coding RNA is expressed in different tissues, okay? And in brackets is the ones that are, it seems to be exclusively expressed in that tissue. So there's not in the other one. So the numbers are pretty low, but there are some of them that seems to be exclusive for each tissue. As I say, that's a very crude analysis. We have to redo that, distinguishing between the sex and between mature and immature. But it gives an idea of the number of long non-coding RNAs we can find there. It seems low compared with the big numbers because, I mean, to be honest, long non-coding RNAs are very lowly expressed. So the expression is very, very low. So the first we tried was to look into similarity. Let's see if we can find long non-coding RNAs are common in all the species. So we did just a normal BLAST10 approach that has been used by many, many authors. Initially, what we use is the zebrafish as a reference, okay, blasted against all the 6 species of the AQUA-FAANG, and it gives us like 148 clusters of the same long non-coding RNA found in the 6 different species. So when we take a look into the similarity, average similarity of all the long non-coding RNAs, we can see that it ranged between 60 and even 100% similarity, but the E-values were not too good. They were not too good. What was happening is that in the majority of cases, we had this situation, that what the BLAST was recognizing was the repetitive regions. So that was a bit of a bummer. So we tried again using salmon, because it has the higher number of long non-coding RNAs, and we used again all the long non-coding RNAs from the other species of the AQUA-FAANG. We had a lower number of clusters. Again, we had repetitive regions that are aligned, so these results are not that good. But in this case, we had a pretty big number of long non-coding RNAs with quite good alignments. So they are potentially conserved between the 6 species. But you have to take into look with the transcriptomic data and structure data to confirm this. We are starting to take a look into the synteny, and initially it doesn't seem to work very well. So it seems that the synteny, at least taking a look at the genomicals, it seems not working very well. There's not conserved synteny so far, or a very poor conservation of the synteny. But again, there's a preliminary analysis. We have to improve this because we've been using genomicals so far. The first look, it doesn't look very well, but we're working on it. The second we were trying to do is, well, let's take a look to the correlations. See, I mean, let's take a look. In this case, we use embryos because you have more time points that you use in tissues because it's not that many. So we found quite 70 long non-coding RNAs that they have very high correlation between the different species, like above 80%. Here's an example of that. Okay, these ones doesn't look to correlate with them, but if you take a look into a more proper scale, they have the similar profile. And what we are doing is a very crude analysis. Now it will be improved with the TAC-seq data and also with the data from the ensemble people that they have this nice promoter detection thing, so it's going to work very well now. But we did a crude analysis of, of the 200 base pairs upstreams of the genes, looking for motifs that were common in all of them. And actually, we found that in the great majority of cases, there were motifs that were 100% conserved between them. So it seems that at least the regulatory regions might be conserved in this case. We will improve that with the data from the other colleagues. So, and again, in this case, we're looking for interactions. That's quite hard, to be honest. It takes an awful long time. We're focusing on 2 kinds of interactions, the long non-coding RNAs versus messenger RNAs, and long non-coding RNAs versus proteins. Okay, in this case, because that takes a long time and it is a very time-consuming process, we were focusing in genes that were related with the muscle because they are, I mean, basically I work with muscle, so for me it's easier to select them. So we were talking about around 500 genes, coding genes. So in this case, for messenger RNA, long non-coded RNAs, we found at least 57 muscle genes that seems to interact with at least 1,800 long non-coded RNAs. That's because we have multiple long non-coded RNAs that can interact with the same messenger RNA. Here is the threshold. That's a program that what they use is they normalize the possible interactions, and it tells you the probability of that the interaction is very strong or not, depending on the free energy. And here's an example with MyoD, a very well-known transcription factor in the muscle. And in BAS, we have 15 long non-coded RNAs that interact, 2 in bream, 2 in turbot, 1 in salmon. I'm very tempted to repeat this analysis in salmon because the numbers were too low. 10 in trout and 9 in carp, with very, very strong predicted interactions. From proteins, we only have done it with Gilchrist bream so far because it takes quite a lot of time, but we're working with the other ones. And in this case, what we found is 32 proteins that are predicted to interact with 39 long non-coding RNAs. The idea is to cross all this data, transcription data, similarity data, and this interaction data to see if there is any common patterns between them. Okay, that's the idea. And finally, because we all know about whole genome duplications, we are trying to find duplicated non-coding RNAs. So in this case, we do just self-blast of the long non-coding RNAs. We look the ones that are highly similar with good A values, and then they have an alignment at over 100 base pairs. And that's the numbers we have found so far, okay, over 150 in all cases, pairs of long non-coding RNAs. And we're looking as well into the conserved structure of the pairs, okay? We didn't finish the trout, salmon, and carp, but so far we found some of them where the structure is very conserved. That's 3 examples of that. Here is a pair of long non-coded RNAs. We compare the secondary structure of the two. Okay, the similarity is quite high, and the structure, if it's under 0.09, that means that they are very similar. Okay, the lower, the more similar they are. And we found this one, for instance, is very, very similar. Is very significant. The correlation between the two in expression, at least in embryos, is very high, and they are located in different chromosomes. So it seems that it could be from a whole genome duplication. It's just an indication. Then we have other ones that they are very similar in structure and everything, but it seems to be originated by local duplications, but they are next to each other. And the correlations are, in this case, they are not positive. They seem to be inverse. But sometimes you think, well, that's because they are similar, but you have situations where similarity is not that high, but the secondary structure is still conserved. So they say that if they have the similar secondary structure, you can be pretty sure that they have the same function. And this is what they say. Okay, that has to be corroborated. And just to finish, because it has to be a very short talk, we are doing something similar with OHNOLOGUES, because I've always been very interested in them. So I'm taking a look into the conservation of the expression between the differences, stages of embryonic development, tissues, sexes, and everything. So we are working on that. And we're taking a look so far if the correlation among species is the same. We can see that some genes have very high, that's the average correlation. I did comparisons between all the genes and all the OHNOLOGUES for all the species, and that's the average correlation of all comparisons. So some genes that have very high correlation, so the correlation is very conserved. Some of them are not that conserved, like in this case. That's for embryos. That's for the body map. In this case, we're taking a look that's only showing the muscle. So we were comparing fold change. Sorry, I'm going very fast, but I only have 10 minutes. So if anyone has a question, I will be very happy to answer after that. And that's the whole list of things we have to do. So we have to finish the analysis. We have to take all the ATAC-seq data. We have to take all the new annotations for promoters and enhancers. And without losing our minds, try to combine everything to make something, that makes sense out of it. Because long non-coding RNAs, so far, they're quite hard to work with. So that's where we are now, and that's where we want to go.